Hello, welcome to lecture 12 of the NPTEL online certification course on Pharmacognosy and Metabolic Engineering. So, today in this lecture we will discuss the biosynthesis of tropane alkaloids. Let us go to the next slide. So, the concepts that will be covered are tropane alkaloid, calistagens, biosynthesis of tropane alkaloids, and we will identify the rate limiting step, so that step can be manipulated leading to any changes in the alkaloid content and composition. And then we will also talk about the discovery of new enzymatic steps of tropane alkaloid. So, this is based on the research work what scientists did for the past 1 decade. Okay. And then we will also discuss about the how to localize the enzymes of this tropane alkaloid pathway in roots. So, because roots are the site of synthesis, I will come to that. So, tropane alkaloids are one of the most important pharmaceutically sought after alkaloids. So, tropane, why it is called tropane? Because it contains a tropane ring. So, this is sometimes called tropane boat. So, that is the active core component of this tropane alkaloid. Now, why tropane alkaloid is important? What are the pharmaceutical importance? I will just tell, but before that I will just let me tell you that hyoscyamine and scopolamine, there is another name of scopolamine is called hyoscine. These are the most sought after pharmaceutically important alkaloids under tropane group. In addition to that, atropine is there, which is the racemic form of hyoscyamine, and then we will also talk about biosynthesis of calistagens, which are not true tropane, but these are called not tropane alkaloids, and we will also see how cocaine is formed, because that also have the has the tropane ring in it. So, let me now tell you the pharmaceutical importance of this tropane alkaloid. For example, the hyoscyamine, what I have shown here, hyoscyamine is basically it acts on the tissue cells and it innervated by post ganglionic cholinergic fiber of the parasympathetic nervous system. So, it is anti-muscarinic and a parasympathetic depressant. Hyoscyamine also has plasmolytic effect on bronchial and intestinal smooth muscles. The mydriatic effect, that means inhibition contraction, which inhibits the contraction of the iris muscle. So, this is also more pronounced than that of the atropine. So, hyoscyamine also reduces salivary and sweat gland secretion, control excess motor activity in gastrointestinal tract. It also producing an antidiarrheal effect, and scopolamine is basically a central nervous system depressant, and it is used for motion sickness, whereas hyoscyamine and atropine are CNS stimulants. So, atropine has been used as an antidote in case of poisoning by cholinesterase inhibitors, for example, physostigmine and so on, as well as this organophosphates. So, if any poisoning happened with these chemicals, so atropines are basically injected, which act as an antidote. So, this is in brief about the pharmacological action of the major alkaloid hyoscyamine, scopolamine as well as atropine. Now, I will also tell you the name of the plants which produces these alkaloids. For example, the atropine is a very common name. So, the plant name is Atropa belladonna. So, in English name it is called deadly nightshade. The other common one which all of you know, Dhutura datura. The scientific name is Datura stramonium and the English name is thorn apple, because the fruit is full of thorns. Hyoscyamus niger which is called henbane, and mandragora officinalis, that is a slight spelling mistake. This is called mandrake, and there is one more plant which is called Duboisia. There are plenty of plants, but these are the plants which are the best sources for tropane alkaloids. Duboisia myoporoides, okay. Now, let us go to the next slide. So, major tropane alkaloid hyoscyamin, which contains the tropane boat, scopolamine, atropine, cocaine, all contains, and calistregin. It is not the true tropane alkaloid, as I said, this is the not tropane alkaloid. Now, we will study how this tropane alkaloids and calistregins are synthesized. So, what is shown here in the left side is a very simplified schematic route for this tropane alkaloid as well as calistregin formation. So, putrescine is basically a polyamine which originates from ornithine, and PMT, putrescine methyl transferase, that is one of the important enzyme which was characterized at the very beginning when scientists they were elucidating the pathways. Subsequently, the gene was also isolated and characterized. Tropine TR1, TR2, one is called tropinone reductase 1, another is called tropinone reductase 2. Now, tropinone reductase 1 basically catalyzing the pathway towards the tropane alkaloid, whereas tropinone reductase 2 basically catalyzing the pathway for calistregins. Now, pharmaceutical importance I have already discussed. So, this slide I basically skip. So, basically this is how the tropane alkaloid is acting at the physiological level, what is showing here. And now, the plant name, this is basically the mandrake, this is the henbane, this is the belladonna, Atropa belladonna, this is dhatura, and this is erythroxylon, which produces the cocaine. Okay. So, as you see that putrescine can originate from arginine, even ornithine also, and then the pathway moves in this direction. Then nicotine is not the tropane alkaloid, but nicotine basically shares the intermediate of tropane alkaloid, which is N-methyl pyrolinum. So, nicotine biosynthesis we will see later in a subsequent class, and here this just now I said that tropinone is the important molecule, because this tropinone reductase 1 and 2 subsequently forms either tropane alkaloid or calistagin. Similarly, from tropinone cocaines are also formed. Now, we will see in brief the biosynthesis of this tropane alkaloid. So, this basically I will now draw here in the board. Ornithine, then as I am using specific color for specific arrows and compounds. So, the arrow I will be joining once I finish writing the name of the compounds. So, what I have written till now are the early steps of this tropane alkaloid formation. There is no tropane alkaloid formed at this stage. So, arginine, by the action of arginine decarboxylase, it makes agmatine. Similarly, ornithine is basically got converted into putrescine, catalyzed by the enzyme which is called ornithine decarboxylase, or ODC in brief, and then agmatine through multiple steps it formed putrescine, and then from putrescine N-methyl putrescine is formed, and the enzyme is putrescine methyl transferase, PMT, just now I have mentioned. So, now we will write the subsequent steps. N-methyl N-methyl pyrrolinium, and then one route goes to nicotine, maybe one route will go to cocaine, and the principal route will goes towards the hygrine, then tropinone, then it makes tropine, okay, and tropinone, okay, and then tropine, then here what we can write, tropic acid, which basically comes from phenyl lactic acid, and that is coming from phenyl pyruvic acid, and that originates from phenyl alanine. Now, let us put the arrow and join it. So, N-methyl putrescine got converted into N-methyl pyrolinum. So, N-methyl pyrolinum, from there its one route goes towards the formation of nicotine. Principal route moves in this direction, it forms hygrine, then from tropinone, then from tropinone to tropine. So, if I put in this way, and then from tropine and tropic acid will join, and tropic acid is coming from phenyl lactic acid, that comes from phenyl pyruvic acid, that comes from phenylalanine. These two joins and make the compound which is what we are concerned with, which is called this hyoscyamine, and hyoscyamine subsequently got converted into 6 beta hydroxy hyoscyamine, and then that finally makes scopolamine. I use a different color to do that. So, the scopolamine is our main compound of interest. So, that is why I put in different color scopolamine. Now, let me add the arrows hyoscyamine to 6 beta hydroxy hyoscyamine to scopolamine, and the enzymes, this is called H6H, this is the same H6H, and then tropine to tropinone to tropine. So, this is TR1, okay, it is 1. Okay. And the other route, what I said, that from tropinone another pathway emerges leading to the formation of callistogens. So, the callistogens I will also put it here, but in a different color. So, from tropine what is called the pseudotropin is formed, PSEUDO, pseudotropin, and from pseudotropin callistogens are formed. So, there are different callistogens. So, like one is the callistogen A3, then maybe the callistogen B2 and callistogen from callistogen A5. So, pathways are move in this direction. So, the tropine to pseudotropin, pseudotropin one moves to callistogen A5, another A3, from A3 it makes B2, from A5 it can also make B2. So, these are the callistogen groups with the not tropine alkaloids. So, they do not have this methylation. So, they cannot be esterified. So, the not the callistogens. So, this is CALYSTEGINE callistogen pathway, and this is the tropine alkaloid biosynthetic pathway. So, this is in brief the basic steps of tropane alkaloid biosynthesis, and this is an oxidase enzyme is there. So, later we will see that this all intermediate steps have been characterized, that we will see later, but first this is in brief an outline of the biosynthetic of tropane alkaloids. Now, let me go to the next slide. So, rate limiting steps. Let us see from the point of product formation. So, the product is scopolamine, scopolamine, and important products are, whereas the other important products in this pathway are hyoscyamine and there is an intermediate which is 6 beta hydroxy hyoscyamine. So, the steps: hyoscyamine makes 6 beta hydroxy hyoscyamine, and from 6 beta hydroxy hyoscyamine scopolamine is formed. Now, the plant species which was used as a model to elucidate the pathways mostly are the hyoscyama species like Hyoscyamus niger, Niger. So, Hyoscyamus niger, that is also belongs to the Solanaceae family, in fact Solanaceae. So, the tropane alkaloids are the characteristic features of Solanaceae family. So, Hyoscyamus niger was used as a model, and scientists they have elucidated the major enzymes of hyoscyamine and scopolamine formation. Okay, so, another now important point to mention here, that tropane alkaloids are synthesized in the root tissues, and from root these alkaloids are transported to the aerial part, that is leaf and stem. So, this transportation happens through the vascular tissue xylem. So, root is the site of synthesis. Therefore, in order to work out the enzymology, scientists they develop the root cultures of Hyoscyamus niger, and as also later hairy root cultures were also developed and used. Okay. Now, as I have determined to tell about the rate limiting steps, why it is so, because what scientists predicted, that if one can modulate this step, then the production of hyoscyamine or scopolamine can be regulated. Okay. Now, the enzyme, as I have mentioned, which converts this is basically the H6H. So, this H6H is basically a bifunctional enzyme. Bifunctional means it has two functions. One is hydroxylase, another is epoxidase. So, this is hydroxylation and this is epoxidation. So, when scientists they characterize the purified H6H, okay, they noted that it is basically the enzyme which converts first hyoscyamine to 6 beta hydroxyhyoscyamine, and then 6 beta hydroxyhyoscyamine got converted into scopolamine. Now, the next question which came to their mind, that how much hydroxylation activity contributes and how much epoxidation activity contributes. So, what they found, very interesting and unusual result, that is the hydroxylation activity is basically contributed by 95% of the enzyme activity, whereas the epoxidation activity which is contributed only by the 5% of the enzyme activity. So, when H6H enzyme is functional, it catalyzes both the steps. First is hydroxylation, which is basically 95% of the total activity, and only 5% is the epoxidation, which makes 6 beta hydroxythiamine to scopolamine. So, this one must remember. So, that actually will help us later to interpret some of the metabolic engineering results. Now, as I have said that I have outlined the basic steps of tropane alkaloids, but I also said that over time there is discovery of the intermediate steps. For example, this one. So, in earlier step I said that N-methyl pyrrolinium converted to tropinone, okay, and there it involves the hygrine. If you see the step shown here. Okay. Now, what we will see here is this, that this is N-methyl pyrrolinium that converted into one intermediate which is called 4,1-methyl 2-pyrrolidinyl 3-oxobutanoic acid. There is a short form of it, anyway we do not have to remember all these things. So, what has been elucidated that from N-methyl pyrrolinium cation by this converts to this intermediate 2 with joining of malonyl CoA, and this is basically a polyketide synthase type enzyme which joins N-methyl pyrrolinium cation and malonyl CoA and forms this intermediate, and this subsequently by the action of cytochrome P450 enzyme, which makes this to tropinone. So, this is a cytochrome P50. So, in other words, this is a new enzyme discovered and this is another group of enzyme discovered. AB stands for Atropa belladonna and DA stands for Datura stramonium. So, in different plant scientists worked and to elucidate the pathway. So, N-methyl pyrrolinium, what I have shown in the very early slides, that it makes hygrine, and what has been now clear that this is basically a spontaneous conversion that can happen, but the other route is basically avoiding hygrine through from N-methyl pyrrolinium through this intermediate by the polyketide synthase, which requires 2 molecules of malonyl CoA, and the next step is basically that a cytochrome P450 enzyme in case of Atropa belladonna, which makes the tropinone, or tropinone reductase is also there, which can also contribute and make this. So, DA stands for Datura stramonium. So, in Datura stramonium tropinone reductase 1, which converts tropinone to tropane. So, polyketide synthase, cytochrome P450 enzyme, and then tropinone reductase 1. So, this is the new discovery. So, now if we see the Robinson synthesis, which was published in 1917 and almost 100 years later, the tropinone biosynthesis has been elucidated at enzymatic level. So, what we can say that the Robinson synthetic chemistry based synthesis of tropinone is now we can say this is what is the biomimetic. So, the original reaction is basically a enzymatic reaction which has been elucidated in 2019. The similar reaction 100 years before biomimicked by Robinson's group synthetically. Okay. So, this underscores the importance of total synthesis in the study of natural product biosynthesis. So, this is very important, and we must recognize the earlier scientist, the organic chemist who elucidated, who basically elucidated the pathway non-enzymatically. So, two more novel enzymes required for Lecture biosynthesis. I have not mentioned Lecture in my earlier scheme. So, Lecture basically comes from phenyl acetyl glucose, which is coming from phenyl lactic acid. So, here is the phenyl lactic acid which I have mentioned. So, from there phenyl lactyl glucose is formed by the action of glucosyl transferase, a glucose molecule is attached, and this then what happened, tropine formed through tropinone reductase. This tropine and this phenyl lactyl glucose will join by the action of a Lecture synthase, and that makes Lecture, and this Lecture by the action of another cytochrome P450 enzyme converted into hyoscyamine aldehyde, and hyoscyamine aldehyde subsequently converted into the hyoscyamine. So, the UGT1 and LS, the Lecture synthase and glucosyl transferase, these are the two novel enzymes subsequently discovered. Now, based on all discovery, let us see what is available. So, this is based on the paper published in New Phytologist in 2020. So, putrescine makes N-methyl putrescine, N-methyl putrescine by the oxidase MPO, methyl putrescine oxidase, which makes this one 4 amino methyl butanol. This is spontaneous, which makes N-methyl pyrrolium cation. That is why straight away in the earlier step it shows from this to this, but there is one intermediate formation and then there is spontaneous conversion. Then what I have mentioned, that this new enzyme polyketide synthase, which makes this intermediate. From there is the cytochrome P50, which makes tropinone. From tropinone to tropane it was known by tropinone reductase 1. So, tropine is ready for joining with the phenyl lactyl glucose. In the meantime, phenylalanine makes the phenyl pyruvate made phenyl lactate, and phenyl lactate now makes phenyl lactyl glucose, and this joins with tropine makes Lecture. Lecture by the action of a cytochrome P50 it makes hyoscyamine aldehyde. This subsequently makes hyoscyamine, and hyoscyamine makes the 6 beta hydroxy hyoscyamine. So, which is now renamed as anisodamine. However, in the early metabolic engineering case studies, I will continue to call it as 6 beta hydroxy hyoscyamine. So, anisodine is basically is a 6 or 7 beta hydroxy hyoscyamine, and this converted into scopolamine. So, but this is catalyzed by the same enzyme H6H and hydroxylation and epoxidation. So, you see this is the hydroxylation, there is no OH here, okay, and this is basically the epoxidation. So, this is more or less the enzymatic route to tropane alkaloid biosynthesis, which is available now with us in 2020. So, now we have come to the last part of our lecture. So, as I said, tropane alkaloids are synthesized in the root. So, the root, if most of you know the anatomy. So, this is the epidermis, outer layer, then there is a cortex, okay, then there is a endodermis, and then that is inside covered with xylem phloem. So, this is the phloem tissue, these are the phloem tissue, and this is the xylem tissue. So, what has been shown that this endodermis is what is shown in the red, which is tissue specific localization of enzyme responsible for this tropane alkaloid. So, this area, that is the endodermis and the pericycle, actually first layer is the endodermis, and this layer, the second layer is pericycle. So, by mistake I cut the pericycle here. So, actually I must write here pericycle. So, so these two layers are very important for this for the localization of the enzymes of tropane alkaloids. Now, this is again a more zoom view of that. So, this is the cortex. So, this is the endodermis. this is the pericycle, and then this is the phloem tissue, all this orange color, and this is the xylem tissue. What has been found that in this ornithine, arginine, so these are there in the phloem tissue, from there it transported here in the pericycle, and then putrescine methyl transferase PMT. So, the ornithine to ornithine formation, ornithine decarboxylase, so ODC will be here, is not it? Yeah. And then this moved to the endodermis, tropinone, the tropinone to tropine. So, that is TR1 or TR2, yeah, TR1, and then the last step which converts hyoscyamin to scopolamine H6H, that is also in the pericycle. So, these pericycle and endodermis are very important, and for this enzymatic conversion. So, in the root, this is the place where these enzymes are localized. So, this has been shown by this PMT promoter fusion with the gas. So, this region is basically the pericycle region where the PMTs are localized. Okay. Similarly, the tropinone reductase are localized here. Endodermis and outside, both here also TR, as well as faint regions are here. Okay, I will try to locate with the yeah. So, this is the region where TR localized, and then this is where is the H6H localized, that is again in the pericycle and endodermis, it is in the pericycle. So, tropinone reductase in the endodermis, and this is in the pericycle, and also the ADC now in the next one. So, this is a very recent publication published in SCS American Chemical Society Synthetic Biology Journal in 2020, where they have shown the expression of Atropa belladonna ODC in the hairy root cell. So, this PC stands for pericycle, EN stands for endodermis, this is cortex. So, the here you see the green the blue, this is basically a fusion of the ODC with the gas, and that is basically localized in the pericycle. Okay, and this is again the another one. So, a maturation zone, this is cell division zone. So, but this is good, and this is also clear here because this is basically a longitudinal view of the root. So, the cylinder, this is the vascular cylinder, that is deep in color blue. So, why? Because that is basically where the genes are expressed. So, that is it is deep blue where lies the endodermis and the pericycle. Okay. So, what I have said that this ODC is localized in the pericycle, and basically this is what is the ODC. So, this is indeed very interesting, and with this latest technologies scientists were able to localize where this pathway is operating. So, with this I end the biosynthesis. Thank you.